development of resistance to ampicillin in Escherichia coli through repeated exposure. So since Alexander Fleming first discovered penicillin in 1928, antibiotics have been used to treat a multitude of different bacterial infections and very effectively. However, over time, the bacteria have been able to develop resistances to these antibiotics, and this is through several different methods. Overexposure to the antibiotic, or non-compliance, which is where a patient doesn't actually take the full course of their antibiotics. So when you go to the doctor, he gives you your pills and he says, all right, make sure you take all of these, and if you actually don't. Resistance can spread between cells, so between generations of cells, and then also between individual cells themselves through methods such as horizontal gene transfer. Also, the movement of people allows for spreading of different types of bacteria as well. So people migrating to different regions, movement of animals to different regions, transportation of meat, anything like that can allow for transmission of bacteria from one region to another, which can also transmit resistance if the bacteria has a resistance. So you can kind of see, I realize it's a little small, but at the bottom of the slide I do have a timeline. So the top portion highlights antibiotic development, so you can see there is definitely a boom between the 1930s and the 1970s. And then lately, obviously it's dropped off. We've only had 2 new antibiotics developed since 2000. And then actually the lower portion of this timeline is the resistances that develop to those specific antibiotics. So you can see as they're being developed, the resistances are being acquired by the bacteria, you know, pretty close. They're very, very close in their shadowing. So the purpose of my thesis was to determine the amount of contact exposure necessary for bacteria to develop a resistance to antibiotic treatment. What I did for this was I took non-pathogenic E. coli from our stocks. Non-pathogenic means it's not disease-causing, it's not dangerous, so you can think of it as an empty suitcase. It's there, but there's nothing bad in it. That's why I was allowed to work with it. I created 3 different bacterial lines. I titled them A, B, and C. I cultured them on tryptic soy agar, that's the TSA. It's a very standard media that we use in the micro lab, so it's not selective for anything, it's just kind of, everybody uses it. Each line I treated with 5 ampicillin discs that had 10 micrograms of ampicillin on them. So I placed the 5 discs in an X-shaped pattern, so 4 at each corner and 1 in the middle, and I did that again for all 3 lines. I then allowed my bacteria to incubate for 24 hours at 37 degrees Celsius. Once the incubation period was finished, I took 7 measurements around each of the discs. So you can see I have highlighted with that little red mark there. I took the measurements from the edge of the ampicillin disc to the edge of the bacterial growth. That's called the zone of inhibition. So I measured from the edge of the zone of inhibition to the edge of the plate. Once I had taken all of my measurements in order to create the next line, the next treatment of bacteria, I took the bacteria that grew closest to the plate. I inoculated those in a 50% tryptic soy broth solution, so it's the same as the agar that I used, except it's a liquid media. I let those sit for a little minute, shook them up, make sure the cells were all, you know, pretty evenly deposited, and then I took 100 microliters of that inoculated solution, and I put those on the tryptic soy agar to create my next treatment group. And I repeated the ampicillin displacement and the incubation, so it was all very strict timing, everything was kept the same. In addition to creating different lines and different treatment groups, I also gram stained all of my bacteria to ensure that all of the bacteria was the same as I progressed through my experiments. So here I have a couple pictures, kind of make it hopefully a little bit easier. So the picture on the far left is actually from my second treatment group. So you can see the zone of inhibition is quite large around all of those ampicillin discs. The middle picture is from my 6th treatment group, and it's kind of shrunken down a little bit. I apologize if they're hard to see. I took all these pictures with my phone, so great camera quality. And then the final picture is from my final treatment group, treatment 13, and it's a little hard to tell, but you can see all of the bacteria is growing straight up to the edge of the ampicillin plate. So to make it a little bit more easy to understand, I have all of my different treatment groups, line A, B, and C, and you can see all of my data from treatment 1 as it progresses to the 13th treatment, so the resistance definitely did develop. Okay, this is also a good demonstration. So once I had developed resistance to the ampicillin plates, or the little tablets, the discs that you saw, to test that, I took all 3 of my lines of bacteria, A, B, and C, and I plated them on a media that was tryptic soy agar, and it had ampicillin already in the agar. So you can see all of my lines of bacteria are growing in the presence of the ampicillin on that plate. And then as a negative control, I had my original untreated bacteria. You can see that's on the right quarter, and it's not growing at all. The agar had 100 milligrams per milliliter, so if you're wondering the concentration there. Also, here's some of my gram staining pictures, just so you can see. The far left is a professional photograph, just because, again, my camera quality was not the best, but you can see the similarities between that photo and the middle photo, which is actually one of my own. So you can see the small little pink rods. They might be a little hard to tell, but they are the same. And I did have one instance of contamination, so that is actually the far right picture. So that's a gram stain of my unknown bacteria, and you can see there's a pretty big difference between my unknown and my E. coli staining. So when it came to this unknown bacteria, I had to mess around in the lab. I had to try to figure out what it was, which is a lot like playing Clue, you know, who did it? Was it in the ballroom with the candlesticks? So there was a lot of work that I had to do in there. I had to plate on differential media, so the far left picture is McConkey agar, and it's selective for certain types of bacteria, so I plated on there. You can see there's no growth. That kind of pointed me in one direction. The middle photograph, I don't know if you can tell, but you can see all of the little pink dots, and then there are also some smaller little green dots. That is an endospore stain. So I did find out that my unknown bacteria was endospore forming, so that also helped push me in the right direction. And then the rightmost picture is the growth pattern of my unknown bacteria. So if you compare it to the pictures that I showed earlier of my E. coli, you can see it's a pretty big difference in the growth pattern. So there's a lot of room for taking my research and kind of pushing it really in any different direction. So I only worked with one antibiotic, so if there's a student that comes on down the line and they want to try doing a therapy that involves multiple antibiotics, so if they want to use ampicillin and then they want to use pretty much any other antibiotic that they might be able to get their hands on and try to develop multiple resistances, you could work with that. I do want to stress that all of my bacteria was disposed of properly. It was autoclaved, which is the standard, you know, how you get rid of everything that you really don't want laying around. And then we did make stocks of my resistant bacteria, so they're in a non-accessible long-term storage, so if any student does want to continue my research, they'll be able to. I would like to thank Dr. Hilwig. She's my advisor, stuck through me the whole way. Dr. Stein, he's one of our micro professors, really nice guy. He actually assisted a lot in my unknown bacteria identification. So we're still not sure what type of bacteria it was. It's likely that it was Bacillus subtilis just because it's a common contaminant, but we didn't have the time or the money to put in to actually figure out what exactly type of bacteria it was. And then also Cheryl Maurer, she's one of our lab assistants. She's really awesome, and she was helping me with my staining techniques. I have all of my references. If anybody wants the list, because I realize it is quite extensive, you can just email me. I'll send that. And we're supposed to hold questions till the end, right? Okay.